Hello, everyone. This is Erick Hernandez. I'm an assistant professor at the University of Maryland School of Medicine. I would like to thank the organizer for this great opportunity. Motility and locomotion are essential for life. Skeletal muscle is a factor of voluntary movements and body balance. It provides bone and joint stability and protects internal organs. Overall, its proper functioning is important for survival and quality of life. Muscles are highly specialized to accomplish delicate, powerful, and complex tasks. Skeletal muscle is the largest organ in the body. It contains over 600 muscles. Muscle tissue is also important for metabolic and endocrine functions. Muscle weakness is a major and common complaint in many acute and chronic disease conditions. Things like Huntington, ALS, myasthenia gravis, diabetes, heart failure, periodic paralysis, muscle dystrophies, and inflammatory myopathies, to name a few, affect skeletal muscle. Yet, in the clinical practice, the wellness of skeletal muscle is commonly overlooked. Hopefully, this trend will change. Nevertheless, this highlights the importance and need of basic muscle physiology research. Skeletal muscle is an excitable tissue. It is composed of large, elongated, multinucleated fibers. Muscle contracts in response to commands initiated in the brain in a process known as excitation-contraction coupling. In excitation-contraction coupling, an action potential is generated at the neuromuscular junction and propagates along the surface and into the transverse tubules. The unique voltage-gated calcium channels, CaV1.1, located primarily on the T-tubule, detects the polarization caused by an action potential and directly activates the intracellular calcium channels to release calcium. This increase in calcium is the signal for contraction in both striated and smooth muscle. At single cell level, this is how an action potential induced contraction and calcium elevation looks. Here's the twitch, and here's the calcium transient. If we could zoom in this region at the T-tubule SR interface, we will see that many proteins and two membrane systems participate in this process, and CaV1.1 is an essential component of excitation-contraction coupling. Today, I would like to share with you results from our current research, which aims to further investigate the mechanisms that link the activation of CaV1.1 with intracellular calcium release. The primary function of CaV1.1 during excitation contraction is to work as a voltage sensor. It is composed by alpha-1, beta, alpha-2 delta, and gamma subunits. The alpha subunit forms the calcium channel pore itself, and it also has the voltage sensing machinery and is the main site of pharmacological modulation. The alpha subunit has 4 highly similar membrane spanning repeats. Each repeat has 6 transmembrane alpha helices, X1 through X6. Helices S5 and X6 contribute to the pore formation. Helices S1 and S4 form the voltage sensor domain. The S4 of each domain has several 4 to 5 positively charged residues. Importantly, each voltage sensor domain is unique on his sequence. So why four voltage sensor domains? Which ones are needed for excitation-contraction coupling? In several members of the voltage-gated ion channel family, individual voltage sensor domains appear to be differentially involved in specific aspects of channel gating. Pioneer studies of the Isakov, Bessanilla, and Olchesi's laboratory have evaluated voltage sensing domain functions of several voltage-gated operated ion channels using voltage clamp fluorometry and heterologous model system. This system offers great advantages and allows for a reductionist approach. Using this approach, it has been shown that voltage-gated sodium channels, domain 1, 2, and 3, are involved in channel activation. In calcium channels 1.2 isoforms, domain 2 and 3 contribute to the channel opening. And more recently, the Chelsea lab has shown that in CaV1.1, BSD1 plays a critical role in calcium current activation. Here, we hypothesize that not all the voltage sensing domains in CaV1.1 contribute equally to excitation contraction coupling. We now introduce a novel approach to monitor conformational changes in each voltage sensor domain during an action potential in adult muscle fibers. Our goal is to track voltage sensor movement in his native membrane environment, where it can interact with other essential and critical regulatory proteins in response to the physiological action potential stimulus. First, we wanted to resolve the time course of the charge movement in response to an action potential. The properties of the intramembrane charge movement, an early event of excitation contraction coupling, have been characterized primarily using voltage clamp step-like depolarization. To electrically monitor the total voltage sensing domain charge move during an action potential, we use the action potential voltage clamp technique. Apply it here to isolated skeletal muscle fibers using the 2-electrode voltage clamp approach. Total current recorded under these conditions was corrected for linear components, given the net nonlinear current in response to the action potential shown here in panel B. During the action potential, the voltage sensor initially moved outward during the depolarization and then reversed during and after the repolarization. Compared to the charge moved by a step depolarization to plus 20 millivolts, which should move the maximum available mobile charge, the action potential moved approximately 2/3 of Qmax. We believe that these are the first support measurements of membrane voltage sensor charge movement during an action potential in a skeletal muscle. Having characterized the charge movement trajectory during an action potential, next, we use functional side-directed fluorometry to track the movement of each voltage sensing domain. First, we utilize side-directed mutagenesis to introduce cysteines at the extracellular region of each S4 in CaV1.1. Then, we electroporate superficial muscle of the mouse, in this case the flexor digitorum brevis, with a plasmid coding for a GFP-tagged and cysteine-engineered calcium channel. After 3 to 5 weeks, FDBs were dissected out, individual fibers were isolated and identified via the GFP expression. Most of these constructs were developed by my life collaborator, Minerva Contreras. Panel A shows the cis-reacting diastane and his correlation with the expression of the GFP and cis-engineered calcium channel, here for domain 2. We use the methanthiosulfonate-based fluorescent dyes, which are rapid reagents for thiol selective modification. In this reaction, the dye is conjugated to the SH group of the cysteine, and to stain the cysteine engineer S4, we depolarize the fibers using either repetitive stimulation or a depolarizing solution. This dye, MTS Tamra, exhibits changes in his fluorescent properties depending on the surrounding environment. Using ultra-high speed line scan confocal imaging, we measured the fluorescent signal in response to a single field stimulus to elicit an action potential and the subsequent voltage sensor domain rearrangement. Panel D, the time course of the BSD movements is shown in a fiber without movement suppression. An early negative signal followed with the application of the stimulus at time 0, and it was shadowed by a slower positive going signal, result of the fiber movement. Movement suppression via BTS eliminates the slow signal, leaving only the early component. in panel E shows the TAMRA signal in a control on transfected fiber, showing the lack of the early signals in response to stimulation at time 0, and the presence of the slow component that can be suppressed with the BTS treatment. Those fluorometric signals from the BSD are likely derived from intrinsic protein rearrangements linked to the S4 domains. Next, we compare the mean normalized fluorescence for each voltage sensor domain with the normalized trajectories of the action potential, the charge movement during an action potential, and the calcium transient and the underlying SR calcium release evoked by an action potential, and measured in a separate set of fibers. Here for BSD1, BSD2, BSD3, and BSD4. As expected, the BSD signal occurred after the action potential. However, their duration and time to peak was quite prolonged and delayed when compared to the measured charge movement during an action potential. Note that the time course of voltage sensing domain 2 accompanied more closely the trajectory of the action potential evoked calcium transient, whereas the rearrangement of voltage sensor domain 1 and 4 were slightly more prolonged, and BSD3 was even more delayed. All the voltage sensing domain signals were also much slower and delayed relative to the calcium release waveform. Thus, a considerable fraction of the signal for each voltage sensing domain could not directly reflect activation of calcium release of charge movement. Yet, a substantial fraction of the signals for BSD1, 2, and 4, but not BSD3, overlap at the racing phase of the charge movement, and even more for the calcium release, and those could reflect voltage sensor rearrangement and or calcium release activation. These experiments were conducted by Dr. Quinterpax, a former PhD student in our lab. Importantly, the system substitutions in each of the voltage sensing domains I've used here in CaV1.1 had minimal effects on the biophysical properties of the gating currents when compared to the wild-type counterparts monitored in this genic myoblast. Likewise, MTS TAMRA labeling produced negligible effects on the kinetics and voltage dependence of the gating currents from cis-modified calcium channels. These crucial experiments in dysgenic myotubes were designed and conducted by Hugo Vivolet, a current PhD student in our lab, and Roger Bannister. Similarly, action potential induced calcium transients here monitored with the calcium indicator in the 1 and the calcium release waveform were not affected by the time staining and repetitive stimulation. In conclusion, the action potential moved approximately 2/3 of Qmax. Part of the fluorescence signal of each voltage sensing domain occurred after the peak of the calcium release and after the peak of the charge movement during an action potential, and thus could not contribute to these events. However, a sizable fraction of the fluorometric signals for BSD1, 2, and 4, but not BSD3, overlap the racing phase of charge movement and even more for calcium release, and thus could be involved in voltage sensor rearrangement for calcium release activation. A clear role for each voltage sensing domain in CaV1.1 in excitation contraction coupling is unclear, but is under current investigation. And we believe that action potential fluorometry can be added to the arsenal of tools used to study voltage sensing domain movements in a skeletal muscle. Next steps. We would like to study BSD movements during repetitive firing and in both slow and fast type muscle fibers using action potential voltage clamp and conventional voltage clamp steps combined with fluorometric tracking. We would like to use unnatural fluorescent amino acid substitutions to track the movement at transmembrane regions in the voltage sensor domains of CaV1.1. These, combined with charge neutralization experiments, will allow us to identify relevant charge residues responsible for excitation-contraction coupling. It will be important also to establish which and how many voltage sensor domains in the tetrads are required to activate the calcium release channel. Finally, action potential ferrometry could be also applied to study other voltage-gated channels in striated muscle, like sodium and chloride. This concludes my presentation. But I would like to thank my mentor, Martin Schneider, for his enormous support. I also would like to thank our group, Quinton Banks, Hugo Babelet, and Minerva Contreras. Daniel Bannett, Roger Bannister, Matt Trudeau, Rich Lovering, Chris Ward, John Leader, and David Weber from UMB for their continuous support and rich discussion. I also would like to thank to our current and past collaborators, especially to Werner Flocker, who provide us the dysgenic cell line. To John Lueck, Werner Melzer, Mark Anderson, Henry Collegraf, and DeVos. I would also like to thank our source of funding and the interdisciplinary program in muscle biology. Thank you all for the attention. And again, thank to the organizers. I will be happy to address questions.